Mononucleosis, also known as mono, is caused by the Epstein-Barr virus and mainly affects MHC class 1. Mono is sometimes referred to as the kissing disease because it is spread through bodily fluids, mostly saliva, but can also be spread through blood and semen as well. Common symptoms include fatigue, fever, rash, and swollen lymph nodes, but can also consist of a swollen liver and a swollen spleen. Because EBV is a virus, it replicates using the lytic cycle within cells and also uses B cells for
So a little
proliferation.
about MHC. MHC allows proteins to be made that help the immune system identify and differentiate between self and non-self cells. Class 1 MHC molecules are actually heterodimers that consist of 3 alpha domain subunits and 1 beta microglobulin. The class 1 MHC has a deeper binding groove than class 2 as a result of its shape, binding peptides 8 to 10 amino acids in length. Processing of antigen into peptides. In an infected cell, constituted proteasomes convert to immunoproteasomes by IFN gamma, which work to degrade the viral protein into peptides that MHC can bind to, forming MHC peptide complex, which is used by CTLs to mount an immune response.
So because mononucleosis is a viral infection, MHC class 1 is a complex that is used to load the antigen peptides. So after the mononucleosis viral proteins enter the proteasome and are broken down into small amino acid sequences called peptides, that's when these peptides are loaded to MHC class 1. And for this to happen, the MHC class 1 heavy chain enters into the endoplasmic reticulum where it binds to calnexin. And calnexin is there to help stabilize that heavy chain. And then after that, we have beta 2 microglobulin binding to MHC class 1, and this allows for the release of calnexin. And this beta 2 microglobulin and calnexin forms a heterodimer, and the heterodimer forms a peptide loading complex with tapicin, calreticulum, TAP, and ERP57. So the job of tapicin is to bring the protein closer to TAP, and then ERP57 and calreticulum interact to protect that alpha domain of MHC class 1 and keep it in the open position to allow for the loading to occur. So once this all happens, then there's a conformational change, which breaks the bond between MHC class 1 and tapicin. And so this final MHC class 1 molecule is now ready to leave the endoplasmic reticulum, or ER. And it leaves in a vesicle, which then is transported all the way to the cell surface. And MHC class 1, bound with the mononucleosis peptide, is then on the cell surface, and it recruits the cytotoxic T cell known as CD8.
Okay, so we have mononucleosis, MHC class 1. And what we have here is that we have the incubation of B cells, and EBV induces an immune response where B cells can capture the EBV antigen, as seen right here, and then it passes it, and then mounts an immune response locally in the tonsils. And B cells can enter circulation and mount an immune response in the lymph nodes, spleen, and liver. We can see that it comes directly in, and this is where the B cells stimulate CD8 T cells, which are important in the suppression of primary EBV infection, which is also important in preventing the reactivation of EBV. We have the B cells also activating CD4 T cells through coach stimulation, which also makes B cells activated, which becomes plasma cells. And plasma cells are antibody-producing cells.
So here we're going to be talking about calnexin, its normal function, and what would happen if it were to have a mutation. So calnexin is a C-type lectin chaperone that retains the bound protein until all subunits relating to the MHC class 1 molecule have folded and associated correctly. Calnexin will release once the molecule has folded, formed disulfide, and the beta 2 microglobulin has attached. So as you can see in the picture, the calnexin is holding on to this part of the MHC class molecule, keeping it bound, allowing for the beta 2 microglobulin to then come in and attach to the rest of the subunit. Once that microglobulin has attached, then the peptide loading complex, along with all of these, will come in and allow for the peptide to then become bound. If there is a mutation on the calnexin, then it may not be able to accurately perform this function of holding it down, allowing for the beta microglobulin to attach. And if that doesn't happen, then most likely the rest of the peptide binding complex will not come in, and then the peptide will not become bound to the MHC class molecule at all.
Okay, so now we will be talking about the other mutation, which is HLA-DM, which stands for human leukocyte antigen DM. Its normal function is that it is a molecular chaperone that works in lysosomes and endosomes of the cells. It protects MHC class 2 molecules from breaking down in its function, and it also regulates how proteins and peptides bind to the mutation. During its function, it will release CLIP, which is CD74 invariant chain protein from the C class 2 molecules, this mutation will also have no effect on the MHC class 1, so it really won't do much to mononucleus disease during its normal function. While the mutation itself, basically what happens when it is mutated, it will result in release of a weakly bound peptide from the MHC class 2 group, and this will then load the peptides that have the higher affinity binding. And normally HLA serum will bind to the N-terminus, so during a mutation, once it binds to the MHC class 2 group, it will then bind to the N-terminus after. A stabled MHC DM will quick load peptides to decrease the chances of peptides degrading, and it will also interact with HLA-DO protein. And then this is our resources, and that's it.